So here we are going to go over genetic variance in meiosis and fertilization. Now, genetic variance is essential because it allows for variability in the gametes, which is going to lead to genetically unique offspring. Now, we're going to talk about 3 different ways genetic variance can arise. Number 1 being crossing over. Now, remember, during meiosis, we have 2 rounds of division. We have meiosis 1 and we have meiosis 2. And in each stage of meiosis, we are undergoing prophase, metaphase, anaphase, and telophase. So these 4 stages are happening 2 times in meiosis, during meiosis 1 and meiosis 2. During that first round of meiosis 1, we have prophase 1. And in prophase 1, the homologous chromosomes are going to come together and they form this unit referred to as a tetrad. Now, tetrad, think of tetra meaning 4 because there's 4 chromatids involved. For example, there's 2 blue chromatids and there's these 2 purple chromatids. So they interact with one another via a process known as synapsis. And they exchange genetic information with one another at specific locations. And those locations are referred to as the chiasmata. Now, these homologous chromosomes are going to exchange genetic information and form these unique chromosomes that are going to increase variability in the gametes. Now, that's exactly what's stated over here. So crossing over, it's going to occur in non-sister chromatids on 2 homologous chromosomes. What are homologous chromosomes? Those are 2 chromosomes that are of similar size, similar genes, and those genes are at specific locations. What are non-sister chromatids? Well, when we take a look at an individual chromosome is made up of 2 chromatids. So this blue chromosome, it's made up of 2 blue chromatids. Those 2 blue chromatids are sister chromatids. But if we took one of those blue chromatids and one of those purple chromatids, we have 2 non-sister chromatids. So the genetic exchange crossing over, it's happening between 2 non-sister chromosomes. So the 2 non-sister chromatids interact with one another on 2 homologous chromosomes via a process known as synapsis, and they exchange genetic information to create genetically unique chromosomes, and this only occurs during prophase 1. This does not occur in prophase 2. Now, another thing to mention regarding genetic exchange is genetic linkage. So this means that genes that are closer together on a chromosome are less likely to be separated during crossing over. If you have 2 genes and they're super close to one another, during crossing over, they're going to move over together. They will not be separated because they are so close together. So genes that are closer together are more likely to be inherited together because they are less likely to be separated during crossing over in prophase 1. Now, moving on, the second way we can have genetic variance is through independent assortment. Independent assortment means that the chromosomes, they align independently of one another during metaphase. So the alignment of one chromosome does not affect the alignment of another chromosome. The alignment is completely random. So the random assortment of homologous chromosomes during metaphase 1 leads to more genetic variance. So we have these chromosomes, and the way that they line up at that midline in metaphase is completely random. We cannot predict where this chromosome will go, or where this chromosome will end up and in which gamete. So it's completely random, and that randomization increases genetic variance. Now, the third way we can have variance is through fertilization itself. Now, fertilization, it's the joining of gametes. So it's the joining of the sperm and it's the joining of the egg. They come together, and we don't know what genetic information each of these gametes is holding. You know, one of those gametes could encode for brown eyes or blue eyes, and the other one could encode for green eyes or black hair or brown hair. Now, since we don't know what the egg gamete and the sperm gamete encode for, it's completely random. That increases genetic variance. Now, those are the 3 ways we can have genetic variance in meiosis and fertilization. It's crossing over, it's independent assortment, and through fertilization itself. and this leads to increased variability and genetically unique offspring.